Okay, guys, so we have been doing a bunch of deep dives into meiosis and how gametes are formed. And remember, gametes are the haploid sex cells that can go and fuse together to form a diploid offspring. And there were some pretty funny things that took place before we really understood genetics. What people thought before this guy Mendel, and I'll show you a picture of him in just a second, but what people thought before Mendel was that genetics were just blended by both parents. So the offspring would be an intermediate of both parents. And so what that really kind of means is like, well, let's use peas as an example since that's what Mendel studied. He said, like, the example of this would be that if a parent were yellow and blue, that's going to be my example, it's poor, then they would have green children. So, like, over here, that's kind of what I have indicated. Blue and yellow would come together to create a green offspring, and that's just whatever kind of offspring you're thinking of. So it's really all thanks to this guy Gregor Mendel. So if you click this link, here's this biography of him. He's this Austrian monk who discovered the basic principles of heredity through his study in pea plants. There's a lot of information on him, but let's just stick to the facts of his work. So he was a pioneer because he was able to obtain, whoops, let's get a better color here. He obtained quantitative data. He also was the first to really have a large number of replicates. So he did this over and over and over with his pea plants. And he was also able to perform 7 crosses. And I'll explain those crosses in just a second. And so that's important because, for one thing, that means that his results were probably reliable. Right? We want to know that what we're understanding is like trustworthy. Can we believe it? Whenever something like an anomaly, right? An anomaly could now be explained. For example, 2 parents with brown eyes having a child with blue eyes. That might have been looked down upon quite a bit before people had a better idea of this genetics stuff. And then finally, we could perform statistics on this. So that we could support our data even more. And what his experiments really showed, so what they showed is that the male, and I'm just going to draw the male symbol here, and female the male and the female, they contribute equally, well, sort of. They contribute equally and what he said was that he also really went into depth, and this is why that brown eye, blue eye thing can come into handy, but the characteristics seen can disappear and reappear after generations. Okay, so that's really important because, like I said, two brown-eyed parents having a blue-eyed child, again, might have really been looked down upon in prior to Mendel's time. So what Mendel did was he took these pea plants and he was able to spend a lot of time fertilizing different pea plants with each other. So he had the first this one standing here, the pea. This stands for the parental generation. And so they typically were homozygous for their traits. Now, it took quite a bit of trial and error for him to figure that out, but the way that he knew it was that, well, he would take a purple flower and he would cross it with a white flower and he would still get a purple flower in that first generation. Okay. So what he was saying when I say homozygous, remember that homozygous, just if we're using, let's say capital F for flower color, actually, let's use purple. Okay, let's use P. So if flower color is P, then this flower might be big P, and this flower here might be, I'm trying to make that little because little p. Okay, so that would be homozygous dominant, and then this would be homozygous recessive. And maybe here on the axial position, right? So this, the flower position, axial would be the dominant one. And so he wanted to make sure that he had a homozygous dominant parent with a homozygous recessive offspring. Homozygous dominant parent and a homozygous recessive parent. Same thing with seed color here. So let's say yellow is the seed color, so big Y, and then maybe little y for green. Same thing here. Seed shape, let's use round or R as our allele. So this seed has the allele for round, both genes, and this seed would have the allele for wrinkled as both genes. And whenever we write these alleles, we write them as lower cases, okay? So that's what he would do. He would isolate and make sure that in his first generation, or in his parental generation, he had homozygous dominant crossing with homozygous recessive. And so he had an F1 generation that formed. So F1 generation. And that's the first round of offspring. Now, he didn't know it yet, but these offspring would have to be heterozygous, right? Because they got one allele from mom and one allele from dad, or one allele from the female flower and one allele from the male flower. And so this offspring would go big P, little p. And this offspring would be big A, little a. Big Y, little y, because they got one allele from each parent. Big R, little r, right? And we can keep going down the line, but those would be our offspring. So now, for him to be able to determine and state that, yes, this is indeed a homozygous dominant, this is homozygous recessive, what he did was he took two of the same flowers and he crossed them for their F2. So he took two F1s, because remember, he has lots and lots of flowers for each generation. So he took two F1s and crossed them together. And so if the F1, let's just stick with the color for now of the flower color, were big P, little p, crossed with big P, little p, this is where our Punnett square work starts to come in. But even without doing Punnett squares, we can distribute these alleles, right? So we know that one offspring is going to be big P, another offspring is going to be big P, little p, and then we come here, right? These two could combine together and we could have a big P, little p, and then finally we could have a little p, little p. And he would see this in his future generations. So then his F2 generation His F2 generation would then present with a 3-to-1 phenotype ratio, which proved that, well, in the F1 generation, they never reproduced to make a white flower. No white flowers in this F1 generation. But in F2, suddenly, white flowers popped back up. And so now what we have is, even though we might have gotten a few offspring that have that recessive allele, remember that that's what kind of started to come together. We don't know what could be passed on. And so if that recessive allele is going to show up, it's going to show up whenever you get a little p, little p. Okay, we'll go over much more of this in Punnett Square talk, but for now, I just want you guys to understand that Mendel really broke the way with identifying that traits could go away and then come back again, and it didn't mean someone had a baby with someone else.